I wonder if we should still give it let's get started. So today's Power Talk is by our Professor Chen, Associate Professor in both our Department of Biomedical Informatics and Data Science and the Department of Medicine in the Nephrology Division. He is also the Associate Chair for Education, although his work is known far beyond the education. Um, and he has a vast background in biomedical informatics. He got his PhD from National University of Singapore and have a tremendous experience as postdoc scholars in various institutions, including at Stanford. He was at the University of Kentucky doing really great work. So we grabbed him from Kentucky, and we're seeing that he's thriving with multiple grants, quite innovative project. This is one of the projects that he's working on. I'm very happy to welcome Jing to talk to us about the large language models in this special domain. Let's welcome.
Thank you, Jake, for the introduction. So today is my honor to present our recent work on protein language modeling. We know that this is really a fast-pacing domain, and there's lots of exciting work. And this could also related to drug discovery or biomaterial preparation. So there's a lot of things could be ongoing in this domain. And most of work of this, most of the job done is by the postdoc Duolin Wang in Dr. Dongxu's lab in University of Missouri. The others were carried by Dr. Qin Shao at the University of Kentucky and also other people. So when we back to the protein engineering, we always want to ask ourselves these questions. What do we already have? And what do we actually want? This is because proteins are very versatile. They have different sizes, different level of dynamics, and the roles could also be very different. There could be catalysts, could be separators, and sometimes binders. We want to learn what this protein can do. We always wanted to put a question mark on them. In these days, with AlphaFold and other technologies, for example, those technology developed by David Baker's lab, we're going to know more about those proteins. But still, if we wanted to go down to the functions or properties of each protein, is not that certain. We always want to ask, say, what exactly are the functions of those proteins in our study? And especially if the protein has a mutation, has methylation, then the structure could change, but that change was not significant sometimes. How does that change lead to change of functions? Is also a question we don't know. We don't know the answer. So many times we want to find a high-dimensional mathematical relationship between the sequence and the function, and that turns out to be a complex problem we want to solve. Protein language model is one way to try to address this problem. This is rooted from a very simple assumption. The sequence itself should have the complete information about that protein. Now, we're not talking about the environment, about the temperature, just say the protein itself. The sequence already have everything. If we can encode the sequence very well, we should be able to know what the exact function should be for those proteins. So this comes idea, say, now we have the language model already, right? We can use a language model to encode the sentences in a book, and then we can let the computer understand what's the meaning of it. They can decode and answer questions, or we can do other tasks. So example we show here is that if you have an amino acid sequence, if we can have an encoder, we can encode this sequence into a vector of some numbers. And then with these numbers, we can put it into a predictor. That could be another deep learning model or or even traditional classification model to predict functions if we have plenty of data and we can train the second deep learning model sufficiently. So that would be the one thing we'll think about. The language model could be utilized to address this problem. This language model could also be used to do a different thing. So you can see in this case, we have amino sequence in as input to the encoder, and similarly, we want to build this vector. But this time, we also fill in conditions, different kind of environment, different the stability problems, and then we wanted to ask this another deep learning model, which is decoder, to combine the information about condition and requirement and the vector to come up a new sequence. This new sequence should be a modification of the previous ones and to satisfy the condition that we specified here. So you can see in the future, we can make new proteins out of existing ones or out of just noise and generate new amino acid sequences to fulfill our needs. So that could be another thing we can do with the protein language model. So you can see what's in fact similar or the concept consistent here is the encoder, because that encoder is kind of considered as enabling technology. Because without that, it's very hard to say what exactly in the sequence. How can we use the sequence? The encoder will convert the amino acid sequence into vector, make it applicable in various conditions. So why this thing, the embodiment is so important? This goes back, in fact, to very old hypothesis called the manifold hypothesis. Because in that idea, we think the real world is high dimensional, has lots of complexities there. But all those things are based on basic samples. For example, in all our physical world, I mean the general daily physical world is a bit you can decipher with Newton's laws. So we can always think about the real world high dimensional data could be related or could be considered resolved using low dimensional manifolds embedded in this high dimensional space. So for the protein language model, we want maybe the same thing. The key is to decode the sequence, to encode the sequence and convert them data from high dimensional space to a low dimensional space and then make it useful for downstream tasks. And there's many similar technologies developed in this philosophy. For example, the principal component analysis is a data projection that would convert high dimensional data in low dimension and make the data usable for downstream analysis. There are many other technologies that all fall into this category. So we want the protein language model, maybe it's also another one that can convert data from high dimensional to low dimension and make them useful. So my first take-home message is that in our real world, the proteins play a crucial role in resolving many of the health challenges. And our task is to infer or predict the functions of the proteins from the sequence. The protein language model could be one of the solutions to address this task, which is to encode the protein sequence into a latent data embedding to make the downstream analysis applicable. That was the first message I wanted to share with you. Then if you look into this domain, you find that this protein language model is not like a new concept. It already has been there for a long time. Starting from 2019, there was a Bowerbird or the Bluebird, and then later on, this will be more specified to different tasks like the KVPLM and also the BioGPT models and so on and so forth. In these models should to be really successful to address their specific problems and push this research forward. And if you look deeper into this protein language models, you found that they are very much like follow the natural language model. They use the transformer model, or here typically is the encoder model, encoder part of the transformer model as their backbone. And they feed in the data. They feed in the sequence data or feed in different biological conditions. And from there, they want to learn more about these proteins. So the comments about there's so many different these protein language models, but their backbones are usually transformer based. This is also easy to understand because the amino acid sequence follows its own order. It's a 1D sequence. The order is important. If you distort the order, you put the amino acid in a totally different order, it's a different protein. So the order of the amino acids could it carry important information, and that can be captured by transformer models, just like the words in the sentences. And that's the same thing we're talking about. So I hope that I deliver the message that's the key to encode, the key to learn about the protein is from the sequence. And to know about the sequence is to embed the sequence into a vector, a latent vector, to facilitate the downstream tasks. So as we can see here, when you have this vector generated, you can put in the decoder to generate a new sequence, or you can put into a predictor to predict the functions of those the proteins. So now we're more focused on how to make this embedding better without training the model from scratch. That's our motivation, because we know that there are lots of protein language models there. There were these encoders being trained several times in different scenarios. How to leverage these encoders to make this work is the key of our research. When we dig deep into this domain, we found that there are several issues. The current embeddings were built based on the language principle. They don't really have the domain revision, which means they don't have the information about how these amino acids will eventually form together to generate a three-dimensional structure, which is more related to the function of those proteins. For example, many of these tasks are related to the mask technology, or MLM, which means if you have amino acid sequence, we just randomly block a few amino acids in the sequence, and we ask the deep learning model to guess what are those missed amino acids. And if they make a wrong guess, then we have a high loss. If they make a correct guess, the loss getting lower. So the model could learn back and forth through the training data about what are the missing letters here. This is biologically related because these amino acids have some revolutionary correlations. So if we can learn the latent revolutionary relationship of these amino acids, we can understand better about this. The whole we can better encode the amino acids into the vectors. So that's the basic assumption in this technology. There's lots of deep learning models developed in this under this discipline. The problem, but you can see here, is that there's no protein structure. We know since the AlphaFold and this AlphaFold 2 and AlphaFold DB, there are lots of structured information available. They may not really say that 100% accurate. There could be some errors here or there, but at least there are information. They could be somehow considered as to help us to learn more about the protein functions. So one of these backbone model we're talking about is ESM model. We have ESM, ESM2, and now the ESM3. So those are foundation models. They were just trained with the mask language model that we showed in the previous slide. And they can represent the protein properties, the stability, the conservation, the mutation efforts. They have been shown tremendous successful in many different domains. They were used as a foundation model, just used as is or fine-tuned for different tasks. But the only thing that you can feel that's missing is the structure, because the structure is really important. And also it's so much directly related with the function of those proteins. Without this structure information, this model are solely sequence-based. Of course, it's useful, but you feel that something is missing. So later on, there are people talk about how about we include this structure as input. So you can see this is another model that we can build the joint embeddings from the sequence and the structure. Of course, it's a simple one, it's abstract one. You can see the top part. You can see here, this was the sequence as input, and it gets encoded. It could be ESM or ESM2 and or 3, then get the sequence embedding. And then the structure, this also structure embedding using graph neural network. You can embed it to build another embedding. And then you can concatenate them into one embedding. That will make sure that this joint embedding has both the sequence information and the structure information. This could be used to maybe address the issues that we talked about, because now you had this structure information. The problem of this is that we don't really have the structure of every protein. This requires dual inputs, right? Not just the first one, just sequence. We know sequence is available. For every protein, we know the sequence. But the structure, not at that level yet. So that could be one problem. Another could be the computational challenges, the slow inference. When you concatenate the embeddings, the size of the vector becomes much longer. So this is my second take-home message. So I want to convince you that to infer the protein functions, we usually would, one of the solutions is the protein language models. But most of the protein language models are trained solely on amino acid sequences. Doesn't mean they are not useful. They are very useful, but this 3D structure information is not incorporated. But we know the function is rely on the 3D structure. So you feel that there was disconnection, and that could make this model less efficient to predict or infer the functions. In the literature, you can see there are two different solutions. One is to enrich the involutionary of the function information in the sequence. But this in the ESM model, for example, but the structure information is not incorporated. The second one is just use both sequence and the structure. But that means the model is bulky, and also the structure information is always available. So that was the problem we are facing now. We have the protein language model. We're not building from scratch, but it's not like structure aware. So this comes out of our solution. So our solution, you can see if you look at the previous diagram, this one is very similar. The only difference is that we don't really want to concatenate the sequence embedding and the structure embedding. We actually say, hey, we have a protein that has sequence and structure. Can you use this structure embedding to revise the sequence embedding to make it so-called structure aware? So that's come as a title we call the structure aware protein language model. In this case, when you train a model, you use both the sequence and the structure. You just say every protein, when they come in, we have the sequence, we have embedding, and have structure has its own embedding. And we like the model learn. Of course, there's lots of technique there, how the model could learn from the structure. But after the model is trained, you don't need the structure anymore. The first encoder one, would learn how to incorporate the structure information. So even if you just provide an amino acid sequence, you can build a so-called structure-aware embedding. To make that could be more useful than sequence-only embedding. That's the basic idea behind this research. To be more specifically, we use a so-called multi-view contrastive learning described on the diagram on the right. So basically, you can see that we have two proteins. We just call protein, but they could be residues or anything. We just call them protein, make it easy to understand. So called C1 is the sequence embedding, and S1 is the corresponding structure embedding. So this C1 and S1 is the same protein. So once these two embeddings are built, since we know that they are from the same protein, we want their distance to be minimized. We want to get closer to each other. Similarly, for the C2 and S2, this is protein number 2. We have this structure embedding, S2, and the sequence embedding. C2, and want them to close to each other as much as possible. Whereas on the other end, since the C1 and the C2 are two different proteins, they should be separated from each other. Similarly, at the structure level, S1 and S2 should be separated from each other. And then the S1 and the C2, these are the different encodings of different proteins. They should be even further apart from each other. Similar is also for the C1 and the S2. So if you think about it, this contrastive learning diagram is about if you have a different views, right? You call the view means different kind of embeddings. You have different views of the same subject. Those views should be put together. They should be similar to each other. And if the same view for different subjects, they should be separated from each other. So that was the idea behind this multi-view contrastive learning. Basically, you put something together and you separate others apart. Contrastive learning is not a new technology. It's been invented a long time ago. So here is just an example show that we have a text, which is a caption, and the images. So when you encode the text and encode the images, if the text and image come together as a caption of the image, they should have higher similarity. Otherwise, it should be fully separate. So this is like a heat map, a distance matrix, which usually can use to help the model learn the correlation between text and the image. And in our case, we use a similar technology, but we wanted to correlate the structure embedding and the sequence embedding. This is a kind of abstract architecture of the protein language model. We have to go to some details about the biology, so you can have this protein structure, and we convert this protein structure into contact maps. And this contact map describe in the 3D space how these amino acids are close to each other. If they are within 22A, we're going to see they are close. Otherwise, they are far away from each other. So we can build this map. And then we use a swing transformer to identify relationships at different scales. And we project this A level, amino acid level embedding to the protein level embedding. With projector is a two-layer, fully connected neural network just for the scaling. And similarly, for the protein sequences, we adopt the foundation model, ESM. And particularly, we use ESM2. And then we encode the amino acid level embedding in the project to protein level. Instead, we have ESM3, but the ESM3 is closed, which means it's not, you can't modify. ESM2 is still open. That's why we use ESM2. And finally, we put these different kinds of embeddings into this contrast learning diagram and let the model to fine-tune the whole network to reach the minimum loss. While doing that, we use the 3 training technologies. One is called the fine-tune top layers, another adopt tuning, another LoRa. We'll talk more about this later. The overall product, final product of this is in fact the sequence embedding here. Not the final product end, is here the amino acid level embedding is the final product. This is embedding of the sequence that's learned from the structure because it goes from here all the way to here, and then we will go back propagation here with tune the A level embedding. And then we can use this for downstream analysis. We can do the protein level clustering, which is unsupervised learning algorithm, and do classification or A level classification. They are supervised learning algorithms. So that's all the downstream analysis, which is independent to the embedding learning technology. We have another version called the SPM2, which was still ongoing. And what we did, the difference here is you can see in the PM1, we use a contact map. But contact map is not the best way to represent the structure of the proteins. In more recent studies, they found that if you have 2 proteins and they put in the 3D coordinates, we can use graph neural network and typically the GVP model to better encode the structure. So we replace this contact map with a structure encoder, a graph neural network based, and then we can do the same contrastive loss and get results. So, and we also show that the PM2 get a better performance than the PM1. And this is the final product we eventually generate. It's ESM2 and the structural modules. And regarding the deep learning neural network, this is inside of the ESM2. And here you can see this is a typical neural transformer network. You have the normalization layer and the attention. And then here we inserted a structure-aware module. And then we go to normalization into this deep neural network and another module. So these two modules here to allow the structure model, the ESM2 model to learn the structure information and in these two steps. And this is very simple. In fact, this is like this is a kind of a compressed model to ensure that we can learn from the backpropagation, the structure information. The backbone of the ESM2 is frozen. We don't change its parameters. And we train this network using Swiss plot library, about 500,000 proteins. And then we use the structure information learned by AlphaFold 2 and develop the code on the Python 1 because PyTorch 1, because this project was started from 2022. And we had the first product 2023 submitted for review, rounds of review 2024. So the review process is even similar to the length of development. That's another funny story. And then we train the model using V100 at the University of Kentucky, the A100 at the Missouri, and the NASCAR Delta, which is really helpful. We got some they'll use CPU GPU time to train it. The batch size is small because the length of the protein and the structure are very bulky and take lots of memory. So we only can handle 20 proteins at each batch. This is just an illustrative example to show that the contrastive learning efforts. So this is a t-SNE plot of the proteins. And these blue points are the structure and this red are the sequence. In the beginning, before contrast learning, they are separate because they are two independent data representation. Then after contrast learning, we pull the proteins together if they are, we put the representation together if they're same protein. So you can see the 10 and 10 here, 15 and 15 here. They are more blended. This representation just to show that the contrast learning can help us put the similar the different views of the same subject together. To make sure I have enough time. So this is another result of the algorithm. Here we use this CKI method to evaluate whether this the algorithms can really separate the different classes. The first row is about the different structure types, the alpha and the beta, and both alpha and the beta. So you can see if you use ESM model, which is a purely sequence-based, it doesn't aware that the difference between alpha and the beta structure difference. So it's all blended together. Well, if you use these other models like a prompt protein and the T5, you still not separate well. This is our PM1, which builds on the contact map. Their separation, they're more separate. And the PM2 is separated even better. And so if you look at the results, you can see this CHR tells how the different classes, different colors separate from each other, while the same class or content. So that shows that this PM2 actually achieve a much better performance than the other models. We repeat this experiment for different architecture and different topology. And from all this, you can see this class, PM2 can really separate the structure info. So just to know that when we do this test, we only provide the sequence. We don't provide the 3D structure information. All the model trained in their own situation, and then we just test them. So the testing data is the sequence only, but still, our model can really separate the clusters made by structures. And there's also other experiment for different kinase groups, and you can see that this is ESM2's results. And some are separate, but you can see this class and this class, they're far away from each other, even though they have the same class label. But in our case, it's better clustered. And then another experiment also shows the better clustering results comparing to the traditional protein language models that solely rely on the sequence information. So this is done that this shows that the unsupervised learning, right? So we get the embeddings and just use embedding to do the clustering, get the results. But what actually we can do is we can fine tune this language model for downstream tasks to make them aware what downstream tasks are there, and then we can feed the embeddings to a supervised learning model to predict the functions of proteins, for example. So here we incorporate three different learning technologies. The one is called fine-tuning. So we know that transformer has multiple layers. In the ESM model, we just keep the bottom layers fixed, frozen, and then we fine-tune the top K layers. It could be the top 3 or top 5 layers to tune. So all those parameters are tunable. The second one is a LoRa, which means that we incorporate this structure, this LoRa learning structure, in the multi-head attention to make sure they are learnable, but other parameters are fixed. And third one is called adaptive tuning. So you can see here, for every structure-aware modules, we incorporated adapters to help it to learn the structure, while all the other components are frozen. So we tried these different fine-tuning technologies, and then we applied the two supervised learning tasks. The first one is the gene ontology network, gene ontology learning. So say if the two proteins are close to each other, they have similar biological process or molecular function or subcellular localization, and then we're gonna say the same class, otherwise in different classes. So we put these different models into this classification task and check the performance. And this is Fmax score. It showed that SPM2 has the best score in the biological process and the molecular function. And the score on the subcellular localization is not the best, but still the second best model here. So overall on the gene ontology, we get a pretty good performance compared to existing models, especially those ESM2 based models. The second one is EC. So EC is a numerical classification for enzymes. And if you have the same EC, just means they have a similar function, similar enzyme function. And here we can see the best model is ESM-based one. So ESM is really good. It's really harder to beat, I would say. So our model is the second best model here in this experiment. We have other experiments on the enzymes, get a more detailed analysis to show that if we fine-tune the PM2 in different technologies, we get a different performance. So if you like to, if you have any specific proteins or something you want to study, or you like to collaborate with us to get further down this road, how use the protein language model for your project, very much like to collaborate. Either you have a wet lab who want to verify the results, or you have interesting protein you want to study, or you have a dry lab, you want more interested in biological problem, want to solve it computationally, well, very much like to collaborate with you because there's so many downstream tasks we can test, we can utilize this protein language models. And we have a PM3, in fact, ongoing. Want to see how that could be used to fit your job. This is just a global picture of the performance of the AI as a protein language model we have at the different geo categories and also different structure information. So here on the bottom, you can see this is about the protein family and the superfamily folding experiments. So my sort of take-home message I like to share with you is that we do see that if we can really build embeddings and then make them three-dimensional aware, we somehow can improve our understanding of this biology by testing on existing knowledge. Whether this could be done for new knowledge, we don't know. We need to find out. We are still on. This is ongoing research. We also implement information on protein 3D structure, and then we make the sequence embedding 3D aware. So this So one thing is we know how exactly to incorporate the 3D information into this sequence-based embedding. But on the other end, when you use the model, you don't need the 3D information because that's 3D embedding or the where is a teaching process. That's only happen during the training phase. Once the model is trained, you don't really need the 3D structure. That means the model could be applied solely on sequence-based technology tasks. I hope that this would convince you that the sequence should have complete information, but to make the 3D aware just to make the embedding better and more useful. I would like to thank the first author, Donglin. Sorry, I didn't really put the name here. The Donglin, she's right now is a research assistant professor at the University of Missouri and did a great work. And Dongxu for leading this project. He's really knows the trend and know what exactly we could do. And Qin Shao, my collaborator and friend in University of Kentucky, he's the driving force, push this forward and get all the resources and make them use for this task. And finally, even this is just one paper and it also take a long time to get published, but we're lucky enough to get our one from NLM, and we have another 3 years to go to further develop research in this direction. And I really wanted to collaborate with you to figure out what exactly this language model can do, what can this do, and how can that help your research? And how can we further advance this language model to fit with your research is our ultimate goal. We are seeking collaborators from all dimensions. Yeah, any feedback or comments or any idea you like to collaborate, very much welcome. I think I will stop here and the questions.
Yeah, very good work. In fact, I'm glad that you're collaborating with Dong Xu, who's a very good friend from 20 years ago, and I still remember, yeah, 19 years ago I was having beer with him in China. So he's one of the most prolific bioinformatics researchers who always innovate, and great job. So one of my first question is about the use of a multi-view contrastive learning. It's very promising and it's much better than single view. I wonder if by using this, we can potentially introduce human bias different from the use case. For example, if you have some view, but then if you ignore other views, but in real world application, people say, I just want one view. Does your large language model, protein language model learned, would be able to adapt to the user's requirement? That's one.
Mm.
And the second is, yes, I agree that in general, the gene ontology fitness is better. But in real world, a lot of the time we're developing drug, there's just involve a single amino acid residue changes that may profoundly change the protein dynamics and the function. Do you have use case? Do you actually systematically look at the cancer mutations and then see whether that will completely separate out the function or there's problem there?
Yeah, I can answer the second question first because that's exactly what we're doing at this time. We work with cancer centers, multiple cancer centers, and particularly on methylation sites, protein methylation sites. Say how that methylation could change the function of the proteins. So one challenge is that the AlphaFold is not sensitive to single nucleotide or single amino acid change. So sometimes when you feed the protein sequence in, you get almost a similar structure. So that's one downside. What we were thinking, as I said, Jake, is to add more views because structure. Why we make them structure aware? Because we know structure is related with the protein. If structure is not the only information that's, for example, methylation is not the only information that relate to function, then we can add other views. The philosophy still stands. Just say how exactly you can encode actual information, knowledge, or data into the multi-view to make it work. And back to your first question about this view, yes, we agree. In fact, when we do this, we notice that not all the protein structure in the AlphaFold 2 or at the same level. Some are very good, very precise, but some are just like crap. So the question is, when you incorporate those low quality protein structure into the model, you in fact introduce noise or bias to a certain extent. So the model, we don't have a paper now, but we're working on that direction. Say how exactly you can infer the potential problem of the different views and address them. This could be either humans input or there could be another the information from a soda source.
Great. Yeah, let's take other questions. John, I see that you turn on the video, so we have plenty of time.
Okay. Yeah, great. I missed the first part of it, so I didn't get all your details on contrastive learning. But one of the challenges with contrastive learning is, of course, picking the negative examples for actually training. And that works really well for a kind of classification problem. So this is kind of what I think Jake was kind of getting at earlier. Have you thought about looking at, maybe joint prediction embedding models, which I've talked to you about before for this type of problem? That may be better for sort of generation tasks and drug repurposing, where you need to generate something new versus using, finding the correct, negative examples.
Yeah, John, I really enjoy your view. I think, yes, contrast learning to a certain extent help, but on the other end, it's also somehow considered to be too simplified. And to incorporate, as you said, downstream tasks could be definitely the angle we can make this job better. And also, I also wondering there's lots of different contrast learning algorithms, but most is distance-based. But the distance to a certain extent is harder to justify. You can say even for vector based, right? What is the distance metric you want to use? And at the protein structure level, if the amino acids are close to each other, they have an interaction. But if they are 28 away from each other, even though they are close, but they may already lose any physical or biological relationship. So that was something we needed to consider how exactly the contrast of learning or other technologies could be used to help us to learn the relation between the structure and the sequence and other information. I mean, contrast is just a path, just the way we do it. If we have a new technology in that domain, and if you're aware of any, we definitely have to collaborate and figure out a better way to build this structure well model.
Thanks. The other thing I noticed too, I don't know if you heard this paper came out recently, they're looking at replacing that entire normalization step with basically a tan H function and getting some kind of good results from that. I mean, that's came out very recently, but when I saw the normalization, I just wanted to mention that.
Yeah, I saw that too. I saw that paper too, from coming his lab, right? So he's talking about what if you don't know and show the similar performance. I think this paradigm shift could change a lot of our architecture, but that's good. That's just make the make us better understand the neural networks.
Thanks. Great talk.
Thank you, John.
Yeah, Chandra, do you have a question to ask?
Yeah, I think this is really cool. I just wondering, do you guys have any source to say because many questions actually is about the talking about interaction among proteins. So I think like a lot of interesting questions in terms of therapy or drug screening is talking about actually how you actually have different effects if you have multiple protein, get the target or those kind of things. So I'm just wondering, have you guys, thought about like extend this framework in terms of protein-protein interaction prediction, all those kind of things?
Yeah. So thank you for the question. I think that's exactly what we are doing and we will do in the following years with this NIH support. We finished this work, in fact, in the early of 2024. So it's almost a year from finishing the paper. It takes a while to get accepted, right? But after that, we already explored many different ways to use this model for downstream tasks as you described, the protein interaction or methylation or ligand protein interaction. Those are all very challenging tasks. But our model is open because what we want to do is build an embedding of the protein. And then the downstream is a different task. It's not like a task specific in our model. So we can make it a little bit separated a little bit from exact tasks. But on the other end, it also makes it difficult to really get this downstream task engaged. Because in one way, if we say we found something, we need a biological experiment verification, and that's we need to find the right collaborator who interested in those proteins, those mutations. And on the other way, we found some collaborator who interested in some proteins, but their results are not good enough if you look at our model's performance. So it's really hard to find a match between the two. So I also wonder in the next step we want to do is do prompt tuning. Because think about the GPT or other models. When you do this model, you don't train them anymore, but you just try to write a good prompt, right? I'm not talking about the cutting edge, those new GPT models. Just say if you use a GPT, you want to write your prompt as good as you can. So if we say our model is say we're going to build embeddings, we should open a port, say what embedding you want to build. Then we can tune our model, or we want not tune, but we just say output different embeddings according to the prompt. So that's kind of prompt tuning technology could be involved here to make it more task specific. That's the direction we are working on. Also the thing is the GSM model, ESM models keep evolving and really fast and get the sequence embedding at another level. I would say already at another level, but they are getting closer. They don't want to open up. They just want us to use it, but not to modify it. That is for our researchers. If we don't modify them, we don't make them structure aware. For example, in this case, what's the point of using it, right? So that's just the caveat in the commercialization and all the and I don't want to talk too much about that, but that's created lots of obstacles.
Thank you. I think because this is really exciting. I think especially I see definitely there's a lot of potential here. I think the tool you guys develop we can actually by using by other people, just like they can build up their specific tools, just like as I said, perhaps I think you guys are more focused on different aspects, but I think just for I actually talking about myself, I just really interested in using this kind of protein language models to looking for different protein complex, all those kind of predictions or all those kind of things. But I agree with you. I think it's really challenging when we're going back to real data, and because all those biological experiments, there's a lot of noise, and actually different tools, molecular tools, experiments, they sometimes give you contradictory results. Even though we already know the ground truth, still, you may not be able to replicate the results. So yeah, I agree with you. I think this is definitely a very challenging aspect when we get into really some downstream applications. Yeah, but still, I'm really excited about this all this development in this direction, because it looks like this protein language model can I think it's more technical issue question I want to ask is that actually depend a lot of resource computationally, or this is just like more, because I know like AlphaFold or other things is really requires a lot of computational resource. So I just wonder if this is something not perhaps this is something language model is more efficient.
This we said that this is already a fine-tuned model, but still it takes lots of computational hours to get it done. So
Okay.
that's a good question. In fact, I didn't say anything about that. The length of protein is sometimes can be 2,000 amino acids, right? And some are short. So this creates a problem when we fit these proteins into the memory. So we have to truncate. We have to use the 512 amino acids. And that's create a problem because if the protein is 2000 amino acids, it just wants 1/4 of it. How can you embed the whole protein? So that's the technical problem we need to resolve in the future work. Regarding the computation resource, we wanted to use existing ESM, we use existing AlphaFold, but we need to fine-tune the models. And to that end, we use resources in our own universities, but also get some national resources. I think there's several chances you can apply for the use of supercomputers. And if they grant you the time, 2,000 hours, you can just use the time for your project.
Okay, thank you, Chen. This is really exciting. Yeah.
Yeah, thank you, Chen.
So related to Chandel's question, so I know that the ESM3 has come out. Does it actually invalidate your work, or you think that you're gonna keep refining other people's model? Yeah, what is the strategy there?
ESM3 and 2, they are both sequence-based. So they just use a sequence as input and solely encode the sequence. Our model incorporates the 3D structures from AlphaFold. But you can see one of the key components we do here. Let me go to that page to see. One of the key components here is we need to modify the structure. We need to modify the structure of ESM2 to make it work. Because if you don't have these learnable layers or the modules, the model can't really learn the structure information efficiently. ESM3 doesn't allow us to make any changes to their architecture. That's their commercial, protection and something really with that. So we have to just stick with ESM2 at this moment.
Yeah, another related question is that there are lots of multimodal that I can't name it, but I remember the one of our UAB CS professor, the new CS professor who's gonna give talk, said that he has a model that has both structure and the protein sequence together. Wonder if there effort to benchmark all of these models.
Yeah.
Yeah,
Yeah, the difference, yes, I can really address this question. The difference is our model is not really, say, multimodal. It's multimodal learning, but not application. So as we showed here, we will have a sequence in. The structure is to help this, to modify this sequence embody, to make it structure aware. Once it was trained, you only need the sequence as input. But this multimodal AI model usually say whatever you use for training, you need when you test it, when you use it, you need to provide the same input. For example, you have image and the captions. You want to use both as input to better know about the image. You train them with both text and image. And then when you use these models, you also need to provide the image and the text. That's what we think is challenging when you want to use in our bioinformatics field because the structure is not always available. That's
Okay.
why we say this structure is only used in training, but not in testing. I think this is the big difference between multimodal AI that we're referring to in the AI world and our strategy.
So my last question, I'm really interested in it, so I would like to ask question. So in the Spark context, and that we want to develop a new antibodies, right? So if we have some novel peptides or yeah, antigen proteins, receptors, we wanted to develop antibodies. Would I actually want to use a more structural or multimodal model, or do you think that your model would be sufficient for me to develop antibodies?
Yeah, I think, Jake, this is a great question. I'd like to collaborate with you further on this topic. I think it was trying. We have to try it first because every protein is different. I can't really say which one's better or worse regarding the models, but I think probably we can try a few and figure out the best strategy.
Okay.
So let's talk more about the collaboration, and also with Dr. Dongshu and Chen. They are great collaborators. They really wanted to get this research ongoing.
Okay, fantastic.
Mm.
I look forward to it. This
Thank you.
is really on the cutting edge, and you've done a fantastic job. Thank you very much. And I think a little advertisement. Jing is organizing ADIS and the 2-day informatics symposium on April 8th and the 9th. There's some last minute call for posters. Right, Jing, do you want to mention that? When is the deadline?
We welcome your poster coming this week or the following week. No matter you have poster, just tell us. We will help you to move forward. But we need now is to know the number of posters so we can prepare for the conference.
Yeah, at least I have 3 posters to contribute. So, okay, all right. I'll see you all, and
Yeah.
bye.